Good morning to all of you. Yesterday, we finished at nucleotide and how does a nucleotide look like structurally. This is what I told you, that in nucleotide, what are the components of nucleotide, how and where it is connected. This is what I told you in nucleotide, what do we have, how it is connected. So now today we are going to start further, and in that we will start with the Watson and Crick model. Watson and Crick model of DNA, and Watson and Crick, they gave the model of DNA. And in 11th also, we have already done the structure of DNA. How does a DNA double helical structure of DNA? How does it look like? So as already I told you, it is double helical. We can make it inside itself. So double helical structure of DNA. It appears somewhat like this. And then we did, as we know, the components of nucleotide. Nucleotide has sugar, it has bases, it has sugar, and it has the phosphoric acid or phosphate. And where it is connected? The nitrogenous bases. The nitrogenous bases are always attached to the first carbon of sugar. First carbon of sugar. And then this sugar is connected at CH2OH end with phosphate. Here it is connected with H3PO4 phosphate. And this results in the release of water. Water molecule is released here when this is formed. So that was the structure of nucleotide which we did yesterday. So how it is, what Watson and Crick, they said that it is always the basis which forms the core of the DNA structure. It is always the basis which forms the internal core of the DNA structure. That means if we have C here, it will be attached by the triple bond to G, and A is attached by a double bond to T, and this way the bases are present in the center on the inner side. And what do we have as backbone of this is, like, then this is connected with the first carbon of sugar. So we have sugar connected here, and here also sugar is connected. To this also sugar is connected, deoxyribose sugar. To this also sugar is connected. And finally, where are they connected? They are connected with phosphate. These are connected with phosphorus, phosphoric acid. Okay, so again, we have in the center one more nitrogenous base. Suppose let's take it G, connected by a triple bond to C, then to a sugar, and this sugar finally attached to phosphate. Okay, this way. So here one more sugar is here. These are connected to phosphorus. And this is how the DNA structure it looks like, connected to always the phosphorus. And we have done this structure when we were in 11th class. Now I told you that there is one more elaborate structure of DNA which we will have to do, and this also was given by Watson and Crick. How does it look like? So that structure I'm going to tell you now. How does it look like? So here, structure of DNA. the more elaborate structure of DNA. Here we have the sugar. So sugar is pentose sugar. This pentose sugar is like this. Okay. Then we have here CH2OH is present here. So I told you to the CH2OH only, this is attached. Okay. This, what is attached? The OH of phosphoric acid, OH. Then here P, OH, OH, and here double bond O. So here it is attached like this, and to the first carbon, here we have oxygen, this is the first carbon, it is attached the bases. Okay, that attachment also I told you how it is attached. That also I told you, if it is a purine, first carbon will be attached to the 9th of purine, and first carbon, if it is a pyrimidine, first carbon will be attached to the first carbon of the pyrimidine. So whichever base you take, suppose we take here C. Okay, so this base cytosine is attached here, suppose, then it will be connected to another phosphorus. Okay, and how it will be connected to the next phosphorus? It will be connected the same way by the, here it will be connected to the O. O. Here double bond P OH, rather OH, not HO. OH, 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 here. Okay, now here, this is here, this, again this to third one, I mean CH2OH of this. So again the sugar is like this. It is attached like this here. Then again the same way to the oxygen. Here another base, suppose let's take that base to be T or A, anything. Then it will be attached by a triple bond to G. It will be attached by a double bond to A. Okay, then another sugar on the other side. And here now again, this will be attached to the oxygen, double bond P, OH, OH, here again OH or HO, anything you can write with your convenience. Now here again this is attached to CH2OH. So here like this, pentose sugar, oxygen here, another nitrogenous base. Suppose we take here A, double bond T. Now this is one side. Okay. Now how it will come on the other side? On the other side, this is G, which will be attached to base. Now how it is attached? The DNA strand is anti-parallel. So for that, if it is 5 prime, 3 to 3 prime, 5 3, 5 3, 5 3, it will be 3 5, 3 5, 3 5, and 3 5. Third carbon first, and then here this is fifth carbon, third carbon. Fifth carbon, see 1, 1, 2, 3, fourth, and fifth. So here it is attached to fifth carbon, then third carbon, fifth carbon, third carbon, fifth carbon, then again here the third carbon. Same way here, because DNA is anti-parallel, so it has to be, if this strand is 5 prime to 3 prime, another will have to be 3 prime to 5 prime. So it has to be 3 prime. For that, the sugar is inverted. So how to make that inverted sugar? It has to be made like this. So this is sugar. So here 3 prime goes up, 3 prime, then 5 prime goes down. Okay. So 5 prime attached to OH. So we'll make here OH, fine, and then P, OH, OH, double bond O. Then again, here, inverted, structure. 3, 5, then again 3. It has to be little down because the shape is not coming good. It has to be little up. So I'm making it like this. So it will be, then again here, then again the connection the same way. So 3 prime, 3 prime, it will come like this. So what do we have? We have anti-parallel DNA. This is what was given by Watson and Crick. Then there was another researcher, his name was Erwin Chargaff, who gave structural evidence of Watson and Crick model. So another researcher, Erwin Chargaff. We have one more researcher, Erwin Structural Evidence. Evidence of Watson and Crick model. Structural evidence of Watson and Crick model was given by Erwin, E-R-W-I-N, Chargaff, E-H-A-R-G-O-F-F. Erwin Chargaff gave the structural evidence, and what was according to him? According to him, he gave few rules. He gave few rules, and what were his rules? His rules were number one rule was, he said that the proportion of A is always equal to T and proportion of G is always equal to C. Proportion of A is always equal to T and G is always equal to C. That means if you describe it, you will say that is A is equal to T, A is equal to T, or you can say A over T is equal to 1. Same way you can say G is equal to C, or you can say G over C is equal to 1. This was the first rule which was given by Erwin Chargaff, and it was the structural evidence of Watson and Crick model. He gave one more rule. The second rule for this was what he said, that there is always an equal proportion of purines. Purines are A and G, which are dicyclic compounds. The proportion of purines is always equal to the proportion of pyrimidines. That is A plus T, G is equal to T plus C. He said that there is always an equal, there is always an equal proportion of purines. What are purines? A and G. These are purines and pyrimidines. When because we are doing DNA here, it's not necessary to write U. Only C and T is okay. C and T is okay because we are not doing RNA that we will use U. So C and T, it is always equal to C and T. That is because we are dealing with DNA. Right now we are dealing with Watson and Crick's model of DNA. So that is, what do we mean by this? That is A plus G is equal to T plus C. A plus G is equal to T plus C, or we can also say that A plus C is equal to G plus T. These all are bases. Adenine plus guanine is equal to thymine plus cytosine. Adenine plus cytosine plus guanine plus thymine. So these two rules were given by by Erwin Shargaf, and these are, you get questions based on this. And along with this, I want to tell you something, that what is the percentage of each nitrogenous bases in human DNA. In human DNA, the proportion of different bases is different. We have 4 bases: adenine, we have say, yes, thymine, we have guanine, and we have cytosine. There are 4 bases which are present in human DNA. Adenine is 30.9%. Adenine is 30.9%. Guanine is 19.9%. The thymine is 29.4%. And the cytosine is 19.8%. So this is the percentage of various this was the I have given you the structure. Base is equal in. Now, after this, I come to another topic. This I'll tell you about the length of DNA. What is the length of DNA? This we have already done once. This is the revision lecture which is going on. And what did in which he said that what DNA is a 2 polynucleotide chain structure. That means in DNA we have 2 chains. I'll erase and tell you. The salient features of DNA structure which was given by Watson and Crick. So, the salient features of double helix DNA. Basically, what did he say? In general, all the salient features. What did he say that DNA, what is basically DNA? DNA is 2 polynucleotide, it is made of 2 polynucleotide chains. What is there in DNA? 2 polynucleotide chains are there. 2 polynucleotide chains. DNA consists of 2 polynucleotide chain, and in this chain, the nitrogenous bases, N2 bases, in this chain, the N2 bases are inside, in the core, inside. When we say inside, that means we are talking of the core. Whereas sugar and phosphate forms the backbone of DNA double helix. See, when I told you that nitrogenous bases are inside. That means nitrogenous bases, what are nitrogenous bases? Guanine is a nitrogenous base. By triple bond to C, here we have sugar, then it is attached to P, this is attached to P, then again sugar, we have another nitrogenous base, by double bond to T, then we have sugar which is connected to P. This way more connections are there. We have here another sugar which is connected to, say we have T, then connected with a double bond to A, then again we have this sugar which is connected to phosphate, P here. So this is the structure. This is the structure, and when Watson and Crick, it is 2 polynucleotide chain. This is one nucleotide, phosphate, sugar, and base. This is one nucleotide. Same way here, base, sugar, phosphate. This is another nucleotide. This way we have so many nucleotides. See, one nucleotide is here. Another nucleotide, phosphate, sugar, base. One nucleotide is here. One nucleotide is here. Here will be phosphate. Here will be phosphate. One nucleotide is this. Another nucleotide is this. So what is DNA? DNA is two. See, one chain is this. Another chain is this. So two polynucleotide chains. Many nucleotides. So many nucleotides. In one nucleotide, one. This is one nucleotide chain. This is another chain having many nucleotides. So two polynucleotide chains. This is what Watson and Crick wanted to say, that DNA is a two polynucleotide chain structure. See, two chains are there. One chain is this, another chain is this. And in every chain, we have many nucleotides. Poly means many nucleotides. See, this chain has this way long chain of polynucleotides. This chain is also long chain of polynucleotides. So what is DNA? It is two polynucleotide chain. DNA is a two polynucleotide chain. And in this chain, if you see, nitrogenous bases are inside. These are nitrogenous bases, which I'll be marking with red color. See, this is a nitrogenous base. These two are nitrogenous bases. These two are nitrogenous bases. These two are nitrogenous bases. So if you see in this polynucleotide chain, the nitrogenous bases are inside. They are inside. They are in the core. And the outer side, the margins, the edges, the margins, the backbone is formed by sugar and phosphate. This is sugar, phosphate, phosphate, sugar, 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 phosphate. So the backbone is formed by sugar, phosphate, and the nitrogenous bases are inside in the core. And it is made up of two polynucleotide chains. This is what he wanted to say. Another thing which Watson and Crick said, he said that both the chains, they run anti-parallel. Two chains have anti-parallel polarity. The second point, the second feature of DNA which he gave, he said that both the chains, they run anti-parallel. This was the first. Second is both strands of DNA, both strands of DNA run anti-parallel. Anti-parallel. What do we mean by this? Their polarity is anti-parallel. That means if we have two strands of DNA, if this is one strand, this is another strand, this is another strand. If this strand, I'll make it with different colors, wait, so that you understand it better. This is suppose one strand of DNA. Okay, suppose here this is another strand of DNA. This is another strand of DNA. Suppose the polarity of red strand is 3' to 5'. The another strand, the black strand, its polarity will be opposite. It will be 5' to 3'. This will be the polarity. This is known as anti-parallel polarity. If first strand is 3' to 5', another strand will be 5' to 3'. This is what he wanted to see. Now another point which he added after antipolarity, he wanted to tell about the bonding, how the various bases are connected to each other, and the type of bonding which is seen in double helix structure of DNA. So third point, he described the type of bond, type of bonding between various components of DNA double helix. What are the types of bonds? See, I have already told you that sugar is there, which is connected to phosphate. Here we have a nitrogenous base, which is connected by a bond to another nitrogenous base, which is again connected to a sugar. Then we have phosphate. Here also we have phosphate. This phosphate is connected to another sugar. Then we have one more base, which is connected to another base, which is connected to sugar, which is connected to phosphate. So this is how they are connected to each other. Okay, this is how the connection is there. This is what DNA structure is all about. Now here, he described the type of bonding. The type of bond which is present in between two bases. This is one base, adenine, and thymine is another base. Cytosine is one base and guanine is another base. So the type of bond present between the bases, these two, this bond is hydrogen bond. Hydrogen bond is present between the bases. Hydrogen bonds are present between the bases. Now, the bases are connected to sugars. If you see this base is connected to this sugar. This base, which is thymine, is connected to deoxyribose sugar. This base, which is cytosine, is connected to another deoxyribose sugar. This base, which is guanine, is connected to another deoxyribose sugar. Now, how they are connected? This bond. What is this bond? This bond, which is the connection between the base and the sugar, deoxyribose sugar, is glycosidic bond. Glycosidic bond. This is glycosidic bond between the base and the sugar. Then we have another bond, which is between sugar and phosphate. The sugar, if you see here, is connected to this phosphate. This sugar is also connected to this phosphate. This sugar is also connected to this phosphate, and this is also connected to this phosphate. Now, what is that bond which is present between sugar and phosphate? The bond which is present between sugar and phosphate, this is phosphoester bond or phosphodiester bond. The double connection here and here with the phosphorus, phosphodiester bond. So phosphodiester bond, because the phosphorus has on two sides, it is connected with sugar. This phosphorus towards this side with this sugar, towards this side with this sugar. So we call it phosphodiester bond. And here, if we see just one connection, phosphoester bond. So these are the three bonds which are present in the structure of DNA. So this was the third salient feature of DNA, which was told by Watson and Crick. And the next, yes, the next was about how, what is the coiling, in which side the coiling is seen, and what about the what about the pitch of the DNA, what about the number of bases in one pitch, what about the length of the DNA strand, what about the distance between the two base pairs, all this. When we say distance between the two base pairs, what do we mean by this? See, this is, I'll write glycosidic bond up because I want to show you the distance between two base pairs. Now this is glycosidic bond, which I am writing here. Glycosidic bond. I've written here. Now, if you see, this is one base, adenine, and this is another base, cytosine. Same way, we can have more bases here. This is one sugar, which is attached to, say, T, and by a double bond, which will be attached to A, then by a single bond to C, then here P, here also P. Okay. Now, there are bases which form the core of DNA molecule. Now, what is the gap between these two? What is the gap between these two? The distance between two bases. What is the gap between these two? This was also told by Watson and Crick, who gave the salient feature of this. And for this, we will have to understand the next point. The next point is, I'll erase this or I'll write on that side so that you understand the fourth point. The fourth point. We are on the fourth point now. The fourth point tells first time that the coiling is in the right-handed fashion. That means if the coiling is there, this is towards the right-handed fashion. Towards right hand. Towards the right hand. This way. It goes towards the right hand. The coiling is towards the right-hand fashion. And then, you know, in this, there is one pitch. What is a pitch? Between this area from here to here. This forms one pitch between the two notches, which we have two notches. This area is known as a pitch. This forms one pitch. Now, In this pitch, see between this, this pitch, the major pitch, the minor pitch, this is the major pitch, which is deep, and this is not deep, this is superficial. So this forms one pitch, and this has 10 base pairs, 10 BP. Base pair is what is the length? How many base pairs are there? This I'm going to tell you now. First of all, I'll tell you what is the gap between the two strands. This is one strand of DNA and this is another strand of DNA. Okay, one strand, another strand of DNA. This gap. I'm going to tell you first. This is about 2 nm. This is about 2 nm, 2 nm nanometer. Or when we write it in angstrom, it will be 20 angstrom. And if we write it in which is 2 nm or 20 angstrom or 2 into 10 raised to the power minus 9 meters. Now, what about this one whole pitch? How much is this pitch? This pitch, one pitch, it is about this one. nm, 3.4 nm, or we can also say 34 angstrom, or we can also say 3.4 into 10 raised to power minus 9 meters. This is one pitch. One pitch is about this. Now, what is the distance between the two base pairs? This we want to take out. How to take out the distance between the two base pairs? This can be taken out by the pitch divided by the number of base pairs. How much is the pitch? The length of the pitch? Length of pitch divided by number of base pairs. Length of pitch divided by number of base pairs. So how much is length of pitch? Length of pitch comes to be about, it comes to be about 0.34. Length of pitch comes to be about 3.4 nm or 3.4 into 10 raised to the power minus 9 divided by 10. Minus 9, it is divided by 10. When we divide it by 10, we get 3.4 divided by 10. We get about 0.34 nm or 3.4 angstrom or 0.34 into 10 raised to power minus 9 meters. This is the gap between the two base pairs. That means it comes to be about 0.34 nm. The gap between the two base pairs. This comes to be about 0.34 nm or 3.4 angstrom or 0.34 into 10 raised to power minus 9 meters. So this was the length of the gap between the two base pairs, which I have written for you, 0.34 nm, or you can say it is 3.4 angstrom or 0.34 into 10 raised to power this meter. So this gap is this much. This gap. What we are talking about, this gap or this gap, you can also write it 3.4 angstrom, or you can also write it 0.34 into 10 raised to power minus 9 meters. The gap between the two base pairs comes to be this much. And how much is the pitch? Pitch is 3.4. And how much is the gap, the distance between the two strands? It is 20 angstrom or 2 nm or 2 into 10 raised to the power minus 9 meters. So this was also given by